Thanks for having me. I am currently working as a researcher at CSANR, Washington State University's Center for Sustaining Agriculture and Natural Resources. But I know some of you from my previous position was postdoc with Deirdre Griffin LaHue's Soil Health Lab. So I'm here to give you sort of the big overview of what I'm seeing in the research on this topic. I'll touch on my my research a little bit, but I wanted to try and give a really big overview of what I'm thinking. And let's see, to start us off, advancing. I want you to think about what word comes to mind when you think of soil microbes. Is that maybe it's the strongest word or the first word that comes to mind? And if you wouldn't mind sharing that in the chat. Just kind of thinking about the context that you have for how you think microbes are important or not important, or yeah. We're seeing bacteria, good, crusty, beneficial, diverse, cycling, shifty, great words. Yeah, and if you think of charismatic little guys in the soil like this image here, then we have a lot of ground to cover, I think, in the next few minutes, because they're very diverse and they are single cells, so they don't probably have a lot of emotions to reactions to management. We'll see some few more. Fungi, potential question mark, misunderstood, ecosystem. Yeah, so there's a lot, it is a hot topic. And it's kind of awesome that the public and everyone is so interested in it. What's interesting for me is my word would be resilient, and I think I'll demonstrate that today, but I'm seeing that they're not super reacting strongly to a lot of management that you think they would. So that's kind of a preview of what's to come here. And I'm going to just, this is a short talk, so I'm going to briefly touch on some studies that look for effects of glyphosate, or Roundup as we know it, on microbial communities, and also tillage effects on microbial communities. And as you're listening, I think, or right now, think about what findings do you expect to hear? And I already give you a hint, but and what do you want to hear? I'm always kind of trying to place new knowledge in the context of what I already know. And I challenge you to sort of think at that level too. Great, thanks for joining me on the exercise. So starting with glyphosate on microbial biomass in particular, I think a study that demonstrates this pretty clearly, even though it's back in 2000, was looking at just adding different doses of glyphosate to soil microbial communities, and 3 days later checking, and they found that there was really no effect of, here on the left axis is the carbon in microbial biomass, and here's the nitrogen in microbial biomass. That's like in these bodies, the cells. And that sort of set the stage for maybe an expectation that there might not be an active site or way for glyphosate to have direct effects on microbial cells. And then pulling in a recent and local study also found some sort of mixed but low effects of glyphosate on soils, and that there was just a lot stronger effect of region, what research farm was being studied at, and a lot of other things besides, and then a very small effect of glyphosate. So I think that's just interesting, as one that people are wondering about. It doesn't mean that other herbicides or pesticides would have no or minimal effect, but this one seems to have a small effect. And this early study even said that glyphosate appeared to be directly and rapidly degraded by microbes, even at high application rates, without adversely affecting the microbial activity. So what might look harmful from someone might look like food for someone else, right? And And this other study found that cropping system, location, year, and root proximity were the primary drivers of the fungal community compositions, and that glyphosate had a small impact on composition and diversity, which I have a graph for, but it was a little complicated. So if you want to see that, let's talk about it later. I want to bring in complicated microbial graphs for this talk. And then just one more that I thought was interesting, because I've known this for a while and I dug back into the literature, but there's a lot of things that can affect microbes, including the higher trophic levels like soil animals. And this study was interesting in that they compared, so the white bars, and in here, I'm just showing the graph with nematode abundance on the y-axis. And their treatments were, the white bars are no treatment, and it was just a field of weeds. And they did a hoeing treatment, and then they did a Roundup application plus hoeing. And there was really no difference between the physical disturbance of hoeing a field and the Roundup application after a few days. And the effect that there was very short-lived. So I think this is another interesting context that they thought to do that physical disturbance as a reference point, which I'm always looking for reference points. Microbes are important, they have functions, but what is that connected to, and what is that relative to, and what is this, how big of a degree? So that's one more graph here, and moving on to the effects of tillage on microbial biomass. And this is a study that is ongoing with other folks at WSU, where we have reintroduced tillage in a field that has been fallow for 9 years, and looked at the effects of a one-time tillage event, no tillage, just reseeded with grass, rototill or power harrow tillage, and looking at the effects of that reintroduction of tillage on microbial biomass was one, but also the weed seed bank and the physical and hydrological properties of the soil. So it was a fun collaboration. And we didn't really see differences in biomass looking at all of the fungal and bacterial biomass, or those two separate. So I did break it down into these funny groups that you'll see on the top of this graph. And you can think of these as different types of microbes. We don't know exactly, can't label functions exactly onto them right now, but there's some ideas. But these are the most abundant of like 30 groups. And you can think of these as like, a really good analogy is cooking oils. So these microbes have different types of lipids in their cell membranes that are structured a little differently, same as your olive oil, your coconut oil, your canola oil. They have slightly different formations in their fat structure. And so we can sort of separate our microbes that way. And so looking at the data in this graph, there are slightly lower biomass numbers for the tillage events, but they're not significant statistically. And there's 8 points per box plot bar here. And these are the most abundant groups. So this is kind of interesting to me. And tillage is one that we do see a variety of responses across studies. I know there's one done by a similar group to that previous paper over near Pullman that found that there were some effects of tillage on microbial composition, but they were also, somewhat subtle compared to what you might expect. And I think there's more here than what we're just looking at. We can talk about later. But a couple of things I'm going to broaden out now and just give some bigger context to these studies. This is a short-term field experiment that I just described, a 2-year cycle. And it seems like the studies that show the strongest effects of any type of management on microbial communities are these long-term experiments, right? Where you're doing continuous corn, or you have cover crops and you're rotating soybeans. And that's the previous work that I was doing in Wisconsin. where I found some interesting effects. So when you are comparing conservation tillage to no tillage, you might not see something, but if you are doing, lots of tillage, that comparison is strong. So the incremental changes are hard to pick up on the effects. And I'll also mention that I've only covered microbial biomass in these few studies. I would say this is somewhat consistent for other measurements we make on microbial communities and I have made, including looking at the diversity of the organisms, the composition, who's there, and different measurements for activity, what they're doing. And diversity in particular is one that surprises me. I rarely see strong changes in diversity, even though we want that to be our proxy for soil health. It doesn't show up in a lot of studies, especially, I'm speaking from my experience too, and that's what I've been seeing in my work and I see often. There's also a couple other types of experiments that we can do to look at the effects of management on microbial communities. And I've worked with a couple large field surveys where we had soil samples from both of the studies are about 120 fields, which is awesome. And you need that because there's so much noise in those data. And still with that many samples, we often see that soil texture and moisture and these other things are overwhelming the ability to detect an effect of the management practices. The ones I'm working right now are potatoes in Washington. And it's been really hard to find anything, even with, it also depends on the range of management practices, right? And the potatoes, I would say, have a fairly small range of management compared to other systems I've worked in. And there's also greenhouse experiments, which are great. And we have gained a lot of our knowledge about microbes and what they're doing from these controlled systems, but then it's really hard to scale that back up to your field, what's actually happening in the field. So I also want to mention that I haven't even touched on this really important topic of what are the effects of different microbes on the outcomes that we're interested in, like yield, or on farm economics, or the quality of the products we're making. And that's because that's just super hard. And I have done a little of that work in a greenhouse experiment and found that there were unique effects of microbes from different farms on corn and rye growth. But again, they were subtle and there wasn't a particular microbial community that was good at decomposing all these different forms of organic nitrogen that we give them. So it makes it hard to know what can we do to manage. So just wrapping up here, a couple take-home messages. You might think this maybe is a little different than talks where people are highlighting their one significant finding. This is bigger view. But I think the bigger picture message is actually hopeful, and it's that microbes are resilient. There's some issues with how we're measuring them, and that these other factors are so more important for distinguishing them. Distinguishing differences in what we're seeing in the microbes, but I think they're doing pretty good. And we can talk about why more. There's some mechanistic reasons, I think, too. And also just reminding you that context matters. I think a lot of the info we're getting on why microbes are so great and helpful is really in isolation, and we have to remember that that's the icing on the cake. They are there. They are abundant. And they're doing their jobs, and it's actually pretty hard. There are a lot of other things that are affecting them more than your management typically. So this is sort of showing that to scale for things that affect microbes, sort of to scale. So with that, I'm just going to leave you with this slide to think about, what did you expect to hear and what did you want to hear, to start thinking about what do you want to talk about now that we're going to move into question and answers.